Okay, so welcome to this short video about the structure of DNA. First we're going to focus on is this image to the right, so I'll zoom in very slightly. This right here is our DNA molecule, and DNA molecule is very often referred to as having a double helix, okay? And a double helix is a structure in which both sides of our molecule have a helical structure, so kind of a twist to it, much like a spring does or a slinky. In between each molecule we have these bases. So if I just highlight them just here, so that will be a set of bases, that will be a set of bases, there, and so on. And these are quite similar to, if you imagine a ladder, in between each side of the ladder you've got these steps. They're very similar. They connect one side of the DNA molecule, one of those helixes, to the other side of the DNA molecule. And that's why I refer to it as a double helix. Really important you use that phrasing because examiners are after using that type of phrasing. Okay. Next thing we want to look at is we want to look at a bit more detail about these bases, so the bit that connect our two sides of the DNA molecule together. In our bases there are three major components. The first component we have is a phosphate group, which is quite often drawn as a circle, just like this. Next in we have our sugar molecule, and then finally on the end here we have our base. Each one of these molecules, if we were to give that a name, we call this a nucleotide. Nucleotide, spelled just like that. So phosphate, sugar, and base all together we call that nucleotide, and if you get a bunch of these nucleotides and you keep them all together, you get the DNA molecule. Within our bases, however, we do have variations. It's not just the same base every time. There are actually 4 types of bases. So if I just get our base back again, there we go. So if we go back here, so we've got our base, wrong pen. There we go. So this here is our base. There are actually 4 types, and the 4 types we have, so we have adenine, which is the first type here, adenine. We also have thymine, then we have cytosine, and finally, spelled that incorrectly, cytosine, and finally we have guanine. And so in our DNA molecule you can either have one of these 4 in this particular bit here, so that there can either be A, T, G, or C, and that's exactly how we refer to it when we are talking about it. So rather than using the whole name, we can just shorten them down to their first letters, so A, T, C, or G. And the other thing you need to be aware about is how they pair. So if we were to imagine on the other side of this molecule, you'd have another base set up just like this, and that other base that would be set up there, so you'd have a sugar group there and a phosphate up here. On the other side of this, it's going to pair up, and that pairing up is only ever going to be in a complementary fashion. So for example, if I were to have adenine on this side, I would have to bind with thymine. Okay, so in our complementary base pairing, we are always going to have A with T, and then alternatively we're going to have C with G. So for example, if I got rid of these 2 and actually I replaced C here, this would have to be a G, and vice versa. It could actually work the other way around. So if there was a G on this side, this would then be a C. If you're struggling how to remember it, the way I always remember it is curly C goes with curly G, and then A and T are both drawn with kind of straight lines, so the straight line letters go together and the curly line letters go together. The last thing we just want to talk about is if, again, let me just paste this down, paste over there, fantastic. if we have our nucleotide and we end up joining onto another nucleotide on the other side, so we've got our nucleotide over here, and we've got our complementary base pairing, so we've got, let's just argument's sake, say A over here and T over here. There is a bond that happens between these two. So between your A bond, between your adenine base and your thymine base, a bond is going to form, and these bonds here are called hydrogen bonds. Okay, and the hydrogen bonds will keep them in place. So if I end up having loads and loads and loads of these molecules, so let's put another one there, for example, and then I can put another one there, for example. You suddenly have loads and loads of bases, and again on each one of these sides you're going to have another base over, oh, wrong tool, you're going to have another base over here, another base over here, again with the phosphate group and the sugar group, and in between each one of these pairings here you are going to have that hydrogen bond forming every time. In the future we'll come on to talk about the number of hydrogen bonds that form, but for now we'll keep it at that. Okay, so just as a quick recap, number one, you have a double helix, and the reason it's double helix is because you have two molecules connected that spin round. Second point we talked about is there are 4 bases, and those 4 bases, A bonds with T and G bonds with C, and that's called, we refer to that as complementary. And finally, the third thing we talked about is between the bases we have hydrogen bonds forming. So hydrogen bonds between bases. Amazing, thank you for listening, and I'll see you next time.